Shigellosis, caused by Shigella, isolated by Shiga in 1896. The main symptom of the Shigella infection is diarrhea, which is bloody diarrhea. That means the stool contains the blood also. Only 100 to 200 organisms are able to cause the infection. Most individuals are infected with Shigella when they ingest the food or water contaminated with human fecal material. Shigella can survive up to 30 days in the milk, cheese, or shrimps. Organism. The Shigella bacteria belong to the family Enterobacteriaceae. It's gram-negative, facultative anaerobe, non-spore forming, non-motile, short rod-shaped, and genetically it is closely related to E. coli. Classification on the basis of their biochemical properties divided into these subgroups. 4 serogroups are there, and these are again differentiated into the serotypes. Coming to the source and transmission of the Shigella infection. These are found in the human intestinal tract, and they can be transmitted by fecal oral route. Other mode of transmission are contact with the infected objects or the sexual contact. Pathogenesis. Main source are man, that is because of the fecal oral route. Mode of the spread is the contaminated fingers, food, flies and fomites. Person to person transmission. Infective dose, that is 10 to 100 viable bacilli, are able to cause the infection. Highest concentration in the stool during the early phase of infection are 10 to the power of 3 to 10 to the power of 9 viable cells per gram of stool. And most convalescent shedding time they have the low count, that is 10 to the power of 2 to 10 to the power of 3. So these are the number of the counts, number of the bacteria which are present per gram of stool. These are during early or the acute infection phase, and this is during the convalescent phase. Coming to the pathogenesis. Shigella cause the bacillary dysentery. Generally, infection is by the ingestion. As we have discussed earlier, very small number of bacilli are capable of initiating the disease. Because, and why it is? Because they survive the gastric acidity better than other enterobacteria. That is why other member of enterobacteria, they require more number of bacteria to initiate the infection, because they cannot survive the acidity of the stomach. But Shigella can survive in the gastric acidity. So that is why a very small number is sufficient to initiate the infection. Now Shigella species generally invade the epithelial lining, and after the invading they will cause the severe inflammation and the death of the cells lining of the colon. The inflammation result in the diarrhea, dysentery, that is the hallmark of the Shigella infection. Toxins are also secreted, that is Shigella toxin 1 and Shigella toxin T2. Okay, the SET1 and SET2. These are the two important these toxins secreted by some of the Shigella species. Now, Shigella dysentery. This strain produces the enterotoxin, that is Shiga toxin. And this Shiga toxin is very similar to the virotoxin that is secreted by the enterohemorrhagic Escherichia coli. Okay. So Shiga and the virotoxin, they are associated with causing potentially fatal hemolytic uremic syndrome. And what is their hemolytic uremic syndrome? When the blood vessels in your kidney become damaged and inflamed. So this is the main role of Shiga toxin, that is similar to the virotoxin in its mechanism. Now coming to the mechanism of pathogenesis of the Shiga toxin. So here you can see this is the intestinal wall. These are the lamina propria. This is the intestinal lumen. Here you can see the M cells. This is inside the cell. So this is the outer area of the intestinal. Now what is happening in the first phase? Bacteria penetrate through the mucosal surface of the colon and invade. So here the bacteria is penetrating and invading the colonic epithelium. But this bacteria will not go inside. It will only divide here in the epithelium cell only. So it will not invade beyond the epithelium into the lamina propria. So bacterial cell will be dividing here only, it will not enter into the lamina propria. Okay. Now here what they will do after the division, they will attach to the M cells of the pair patches. So here they have attached to the M cells of the pair patches. Then after attaching to the M cells, they will transport inside the intestinal lumen, and they will be going to form and attaching to the macrophages. So M cell typically transport the foreign antigen from the intestine to the underlying macrophages. So from here they will go and they will transport to the macrophages which are present inside the intestinal lumen, that is in the lamina propria. Now here Shigella will lyse the phagocytic vacuole and replicate inside the cell, inside the cytoplasm. Okay, so this way here we have seen there are the different steps. Now we will read it here that bacteria penetrate through the mucosal surface of the colon and invade, and they will multiply the colonic epithelium, but they do not typically invade beyond the epithelium into the lamina propria. So this is the first step, that they are here. Then they will attach to the M cells in the Peyer's patches of the small intestine. So M cells are the lymphoid cells, lymphoid tissues. Now M cells, they will transport the foreign antigen, that is the bacteria, from the intestine to the underlying macrophages. So these bacteria, these antigens, they will be transporting into the macrophages. Then the Shigella can lyse the phagocytic vacuole and they will replicate into the cytoplasm. And as a result of that, the inflammation of the cell will take place. Inflammation of the intestinal lumen will take place. Now the clinical features after this action of the bacteria inside the intestinal lumen, as a result, different chemical features will appear. So these are fever, bloody diarrhea, mucus and pus in the stool, tenesmus, that is the cramping rectal pain, then convulsions, that is uncontrollable muscular contraction. Now coming to the causes of infection. As it is transmitted by the fecal oral route, so because it is a bacteria of Enterobacteriaceae family, it resides in the human intestine. So we have the causes of infection will also be the unhygienic practices, eat the contaminated food or the contaminated water. So these are the cause of infection. Now there are certain risk factors also, like the children they are more likely to get the infection. If a person is living in such area like very highly populated where the sanitary practices are not good, or it is exposed for longer period of the time in the improper sanitation condition, so those are exposed to the infection, and these are also very important risk factor. Now complication. Generally Shigella infection clears up without any complication, but infection will be clear up, but sometime it may take weeks or months before your bowel habits return into the normal. Sometimes complication can arise. So what are these complications? These could be dehydration. So persistent diarrhea can cause the dehydration. So the symptoms for this dehydration will be lightheadedness, dizziness, lack of tears in the children, sunken eyes, dry diapers, and severe dehydration can lead to the shock and death also. Second complication is the seizures. So some children who run high fever with the Shigella infection can have seizures also, that are the convulsions. Rectal prolapse, that is also there, that is in this condition straining during the bowel movement may cause the mucous membrane of the rectum to move through the anus. So that movement of the rectum that will move towards the through the anus. So that is very significant complication. Fourth is a hemolytic uremic syndrome. So that we have seen earlier also, that they are low blood count is there, that is hemolytic because of the damage of the RBCs, and kidney failure is also there. Okay. Toxic megacolon. So this rare complication occurs when the colon become paralyzed. So bowel movement will be prevented. So that is the toxic megacolon. Reactive arthritis. So that is also developed in response to the infection. Okay, so here what will happen? The inflammation at the ankle, knees, feet, hips, all these places, the inflammation will be there. Coming to the prevention. So washing the hands frequently and thoroughly, supervise the children, dispose of the soiled diapers properly, disinfect the areas, do not prepare the food for others if you have diarrhea, so that we can prevent the infection. Keep children with the diarrhea from so all the things, avoid soiling water from the ponds, lake. So all the practices should be adopted to prevent, for the prevention, in which the proper sanitary practices should be followed. Coming to the lab diagnosis. So lab diagnosis, here we will cultivate the bacteria, that is from the fecal matter. Feces will be the this specimen, and here we will culture them on the specialized media like McConkey, DCA, desoxycholate agar, and XLD xylolysine dextrose agar, and selenite broth. And then we will go for certain biochemical test. So if we are like MR positive, nitrate reduction positive, oxidase negative. So these are the certain significant lab diagnosis by which we can identify or diagnose the Shigella present in the sample.